In this talk, we will learn about the classification of fungi, which are eukaryotic organisms. Eukaryotes are thought to have evolved after prokaryotes. According to the fossil record, we are able to detect single-celled eukaryotes around 2 billion years ago. Now, U stands for true and karyon means nucleus or kernel. Now, nucleus itself means a tiny kernel or a nut. The reason the nucleus was named this way because when we look at a cell and observe the nucleus, it does indeed look like a tiny nut or kernel that is present in the cell. The nucleus is the compartment of the cell where the DNA is present. In the case of all eukaryotes, we do observe the presence of the nucleus, which is the home of its genetic information, or DNA. All eukaryotes are grouped in the domain of eukarya. There is immense diversity among eukaryotes. Some are unicellular, while others are multicellular. There are 4 traditional kingdoms in the domain of eukarya. The first one is Protista. The organisms that belong to this kingdom that has now been abandoned are protists. The reason it has been abandoned is because the members are genetically very variable from one another, and hence this kingdom is not a monophyletic group. Fungi is another kingdom of eukarya, followed by Plantae, which includes organisms that are photoautotrophs. Animalia is the last or 4th traditional kingdom that is observed in the domain of eukarya. Fungus, which is the singular form of fungi, originates from the Greek word of sphangos, which means sponge. This is based on the sponge-like appearance of some mushrooms that were present at that time. Members of the kingdom of fungi belong to the superfamily clade of Opisthokonta. Opisthokonta, in turn, is a member of the clade Obozoa, which is part of the superfamily of Amorphia. It is interesting to note that animals also belong to the clade of Opisthokonta. It is estimated that fungi evolved over 1 billion years ago. We do have fossils that show that they were present for at least 500 million years, but more evidence is needed in terms of fossils to show that they were present even earlier than that. By performing DNA sequencing, it has been shown that fungi are closely related to a group of protists called as nuclearides. These are unicellular aquatic protists. However, we do know that there are many fungi that grow on land, and hence it is considered that they are the early organisms to colonize land. It's not clear how many fungi species are present, but based on metagenomics, it is estimated that there are at least 1.5 million species that are present currently on Earth. Some fungi are unicellular, and a perfect example of these are yeast. Most fungi, on the other hand, are multicellular. They tend to show the appearance of tube-like structures which resembles filaments, and hence they're also called as filamentous fungi. There are a few fungi that are dimorphic, which means that in some conditions they exist as unicellular yeast, while in other conditions they can change and adopt the structure of multicellular fungi and show the presence of the filamentous form. Some common examples of filamentous fungi are mold and mushrooms. Filamentous fungi show the presence of tube-like extensions of cytoplasm that are covered with the cell wall. These tube-like extensions are called as hyphae. There are different types of hyphae. The first one is where each hypha shows the presence of a cross-wall or a septum. Such hyphae are called as septate hyphae because of the presence of the septum. The septum divides the hyphae into cells, and in each cell a nucleus is present. Now, the septa, or these cross walls, tend to have pores and hence are porous. It allows the cell content of one cell to mix with the content with its neighboring cells. This includes exchanging proteins as well as ribosomes. Another type of hyphae is the one that do not have cross walls, and hence there are no septa. In this instant, each hypha appears as one single cell with many nuclei. This condition is called as cenocytic, and hence these hyphae can be called as aseptate, non-septate, or cenocytic hyphae. In recent years, many fungal genomes have been sequenced through a powerful technique called metagenomics, which does not require scientists to grow the organism in large numbers. With the help of metagenomics and the sequences thus obtained, scientists have been able to build phylogenetic trees like the one shown in this figure in order to determine the evolutionary relationships among different fungi. This classification is being revised periodically. For example, the phylogenetic tree that we are able to see here is from the early 2000s. Fast forward 20 years, and now we're able to see another phylogenetic tree that includes many more members. We're able to see a huge group called as Ascomycota and another group called Basidiomycota. As time progresses and we get more sequence information, do expect to see more changes. For the sake of simplicity, let us look at the phylogenetic tree that was proposed in the early 2000s. When we look at the different groups that comprise of different fungal members, we notice that with the exception of one, all of them have the word mycota at the end. Now, in Latin, mycetes means fungus, while in Greek, myx or mycet also means fungus. Thus, mycota derives from the Greek word for fungus. Now, there are certain types of groups that do have the word mycetes or mycota in them and are fungi-like, but they do not belong to the kingdom of fungi. These include actinomycetes, which are actually bacteria and hence are prokaryotes, oomycetes, which are protists, and slime molds, which are also protists. In this talk, we will look at the members that belong to the groups of Basidiomycota, Ascomycota, Mucoromycota, and Chytridiomycota. Members of the group Chytridiomycota are also known as chytrids, and research has shown that they diverge very early in fungal evolution. Chytrids are found in diverse habitats that includes water bodies and soil. They make these spores that are very characteristic in that they have flagella. These spores, called as zoospores, are present in a pouch. In fact, chytrid comes from a Greek word that means little pot that is indicative of this pouch in which the zoospores are present. Recently, chytrids have been responsible for the worldwide decline in various amphibian populations. They cause a disease in the skin of the amphibians, and we're able to see the presence of those pouches that have the zoospores present in them that allowed scientists to determine that this disease was indeed caused by chytrids. Scientists have even found out the causative agent of this disease called as chytridiomycosis, and the agent is named as Batrachochytrium dendrobatitis. Glomeromycetes are fungi that are members of the group Mucoromycota. These organisms have multinucleate hyphae that lack septa, and hence they are cenocytic in nature. Glomeromycetes reproduce asexually, and there is not much evidence to show that they undergo sexual reproduction despite having genes that help with meiosis. Glomeromycetes are known to form symbiotic relationships with plants. Such fungi are called as mycorrhizae. Myco means fungus and rhizome means root, and thus these fungi are able to associate with the roots of plants. There are two main types of mycorrhizae, which are the arbuscule mycorrhizae and the ectomycorrhizae. Most glomeromycetes are arbuscular mycorrhizae. Their fungi extend the hyphae through the cell wall of the plant cells that form the roots. Even though they're able to penetrate through the plant root, they do not extend into the plant's cytoplasm and are covered by the plasma membrane of the plant cell. Here we're able to see another picture where multiple arbuscules are formed in different cells of the roots of the plant. The arbuscular mycorrhizae are able to provide the plant with minerals and water. They help the plant become more tolerant to droughts or deficiencies of water, and they also help them become healthier. In return, the plant provides the fungi with nutrients. There is evidence that suggests that arbuscular mycorrhizae helped the early plants to survive the infertile soils that were present then and hence colonize the land better. In fact, when we look at plant roots that have mycorrhizae associated with them, these roots tend to grow better and are healthier. Mycorrhizae also help make soil. The way they do that is they release enzymes into the environment, which is usually the land in which they live. These enzymes that are released by the hyphal cells can then break down complex material into simpler material. This simpler material is then absorbed by the hyphae as nutrients. Thus, they're able to loosen up different types of components present in land to form soil. Mucoromycetes are another member of the Mucoromycota group. These fungi are multinucleate and their hyphae lack septa and hence are cenocytic. They can reproduce asexually as well as sexually, and a common example of a mucoromycete is Rhizopus, which is known to form mold on bread. Rhizopus can undergo asexual reproduction where specialized aerial hyphae are formed that are called as sporangiophores. At the tips of the sporangiophores are sporangia, and within the sporangia are spores. These haploid spores are genetically identical to the parent plant. During sexual reproduction, a specialized structure called as a zygosporangium is formed. Within this zygosporangium, spores are produced which are called as zygospores. When these spores are released into the environment, they can then germinate to give rise to the hyphae and the appropriate mycelia. Unlike what we have seen so far, members of the clade Dicaria have septate mycelia, which means that we are able to see septum or cross walls present in their hyphae. The members that belong to this clade show the presence of dikaryotic mycelia. This type of mycelia is formed during sexual reproduction. This process occurs when mycelia of different mating types are able to release pheromones. These pheromones are chemicals that draw the mycelia towards one another, and thereby they're able to meet. After a compatibility check, these hyphae are able to fuse. As their mycelia fuse, cytoplasmic mixing can happen. As the two different mycelia fuse to form one mycelium, the cytoplasmic contents mix, but the nuclei can remain separate and not fuse. Such mycelia is called as heterokaryotic mycelia. Within this heterokaryotic mycelia, in the case of septate fungi, the septum starts being generated. This results in different cells. However, in each cell, two nuclei are present, one from each type of mycelium. And hence this type of mycelia is called as dikaryotic mycelia. In clade Dicaria, we are able to observe the presence of this dikaryotic mycelia. The nuclei can remain separate for an extended period of time until meiosis occurs. Members that belong to the clade Dicaria include Ascomycota as well as Basidiomycota. Ascomycota is a group that contains about 75% of the known fungi. Hence, it is a very diverse group that includes bread yeasts like baker's yeast, common molds, cup fungi, truffles, morels, etc. It thus includes unicellular as well as multicellular fungi. Ascomycetes are sometimes commonly called as sac fungi as well. They get their name because they produce these fruiting bodies called as ascocarps. Within the ascocarps, there are sac-like asci, and within these asci are spores that are generated through the process of sexual reproduction and are called as ascospores. For the formation of ascospores, the first step is the fusion of mycelia. Mycelia from different mating types produce chemicals that allow them to move towards one another. Ultimately, they will fuse to form a single mycelium, where the cytoplasm from each mycelium mixes. This process is called as plasmogamy. After plasmogamy has occurred, we will have the formation of a septum. This septum will then lead to the formation of dikaryotic hyphae. Now within the dikaryotic hyphae, we will also see the formation of an ascus. This ascus is actually formed in a fruiting body called as the ascocarp. The nuclei within the ascus will then fuse to form a genuinely diploid nucleus. This process is called karyogamy, and the diploid nucleus formed is similar to a zygote. Following karyogamy, meiosis will occur, which leads to the generation of 4 haploid ascospores within the ascus. These ascospores are genetically different from each other as well as their parents, and hence sexual reproduction results in genetic variation. A mitosis event then occurs, which then results in 8 haploid ascospores that are present in each ascus. With time, the ascus will mature and the spores will then be dispersed from the ascocarp. The spores that are dispersed will then germinate to form hyphae and mycelia. Thus, this is the whole process of the formation of ascospores. In addition to sexual reproduction, Ascomycetes can also reproduce asexually by forming asexual haploid spores called as conidia. These conidia are produced at the tips of specialized hyphae that are called as conidiophores. The conidia that are thus generated and dispersed can germinate to form hyphae. These hyphae will then grow to form a network of mycelia. It should be noted that the conidia that are thus produced are genetically identical to the parent. Hence, it is a form of asexual reproduction. Ascomycetes have huge impacts on the environment as well as in our lives. Some ascomycetes form lichens, which are symbiotic associations between the fungus and green algae or cyanobacteria. Thus, a lichen is an association between a fungus and a photosynthetic microbe. Lichens are found everywhere. We can see them on tree trunks as well as on rocks. They vary in size as well as in color. The fungi is able to protect its photosynthetic partners from strong light as well as from drying out. Lichens are very resilient and have invaded even the harshest habitats. They are sensitive to pollutants and hence can be used as indicators of how polluted an environment is. Some ascomycetes are used in the food industry to make bread, cheese, and other types of products. These include yeast as well as certain types of molds. They are known to cause diseases in crops and thus are responsible for the loss of many types of fruits and vegetables. Some species of Ascomycetes can infect animals too. Candida species are known to infect humans, while Ophiocordyceps species are known to infect different types of insects like ants as well as spiders. In fact, in the case of insects, Ophiocordyceps is able to change their behavior, and thus these insects get the name of zombie ants or zombie spiders. It is in fact the inspiration for the popular video game and show called as The Last of Us. Some species of Ascomyces are also source of antibiotics that are commonly used to treat bacterial infections. Some commonly used antibiotics are penicillin and cephalosporins that are obtained from different types of Ascomyces. Another member of clade Dicaria is Basidiomycota. These include fungi like mushrooms, toadstools, puffballs, etc. They possess septate mycelia and hence are not cenocytic in nature. They get their name because they produce fruiting bodies that look like pedestals, and hence the name basidiocarps. Examples of these fruiting bodies include mushrooms. Thus, any of these aerial mushroom-like structures that we see are actually fruiting bodies. The basidiocarps generate spores called as basidiospores that are obtained through the process of sexual reproduction. The formation of basidiospores begins when haploid mycelia of different mating types secrete chemicals and then come close to one another. After a compatibility check, the mycelia will fuse and their cytoplasm mixes in the process of plasmogamy. The nuclei, however, do not fuse and remain separate. Ultimately, septa are formed and dikaryotic mycelia are formed where in each cell there are two nuclei. The fungus can remain in this dikaryotic state for a long period of time. When the conditions are right, a basidiocarp is formed. Within the basidiocarp, we have basidia, and in the basidia, we still have the cells which have two nuclei. Ultimately, karyogamy occurs in the basidia, and as a result of this karyogamy, where the two nuclei will fuse, we end up with basidia. They have diploid nuclei. After karyogamy, meiosis will occur, which will generate the formation of 4 haploid nuclei. These 4 haploid nuclei will eventually form basidiospores, and thus in each basidium we have 4 basidiospores. These basidiospores are genetically different than the parent fungi. Ultimately, these basidiospores will be dispersed into the environment, and when the conditions are right, these spores will germinate to give rise to hyphae, which eventually will form mycelia. Some basidiomycetes form associations with plant roots called ectomycorrhizae. These ectomycorrhizae do not penetrate into the root cells, but surround the root cells. This is different than the arbuscular mycorrhizae, which are able to penetrate into the cells. This can also be seen in the other figure, where the ectomycorrhizae are able to weave themselves around the plant cells that form the roots, while the endomycorrhizae, which is another name for the arbuscular mycorrhizae, are able to penetrate into the actual cells that form the root. Some basidiomycetes are known to cause diseases in plants like different types of rust and smuts. Other types of basidiomycetes are known to be poisonous, like the deathcap mushroom. There are some others that are known to make hallucinogens and hence are used to make psychedelic drugs like psilocybin. With this, we come to the end of our talk where we learned about the origins of fungi and different features of fungi. We also looked at the classification of fungi and were able to learn about some members present in different groups of fungi. We also looked at the impact of fungi on the environment as well as on humans.